Okay, thank you. Next, I'd like to invite Dr. Jose Lopez from our Division of Hematology Medicine at the University of Washington, who's going to give his presentation on an in vitro system for the study of small blood vessels. Jose?
Let's start my own timer here. So thank you very much for the invitation to come here. I show this first slide for two reasons. The first is to point out a couple of people who are both in the room, Ying Zheng, who developed the system as she was a postdoc. She developed the system as a postdoc and has continued to refine it. And Junmei Chen, who's on my lab, who's done a lot of the work that I'll talk about. So, and the other reason is because usually at this time I'm backpacking in the Cascades, and this is Tank Lakes in the Alpine Lakes Wilderness, just east of Seattle. So the topics, I'll talk a little bit about the endothelial contribution to thrombosis at the level of small blood vessels, the role of flow and vessel diameter and vessel geometry in these phenomena, and use this opportunity to describe this in vitro vessel system for the study of angiogenesis, barrier function, and tissue modeling. So many diseases are characterized by small blood vessel occlusion, and these include thrombotic microangiopathies, and that includes thrombotic thrombocytopenic purpura, or TTP, hemolytic uremic syndrome, but there are others like malaria, sickle cell disease, and sepsis. So as a model, thrombotic thrombocytopenic purpura, a rare hematologic disease that'll kill you if you get it and don't treat it. And it's characterized by low platelet counts, mechanical red cell destruction, when there is no alternative explanation. And what one finds is systemic microvascular platelet thrombi. And if you look at a normal blood smear, what you have nice red cells, and you have a few platelets around the smear. In a patient with TTP, what you have in this particular smear are no platelets and a bunch of shredded, mechanically damaged red cells. And here's just a consequence of that is that there's small blood vessel occlusion. Here's a glomerulus shown. So a few years back, my colleagues and I discovered that a finding that might shed light onto the mechanism of TTP. And that was that if we activated endothelial cells and perfused platelets over them in the absence of plasma, that the activated endothelial cells would bind the platelets and form these beads on a string-like structure. And what we subsequently learned that what it was that the endothelial cells secreted von Willebrand factor. The von Willebrand factor individual molecules, this is a large multimeric molecule, they can self-associate to make essentially endless strands that orient in the direction of flow, and those strands bind platelets. Normally, these strands are removed from the endothelial surface by the action of a metalloprotease named ADAMTS13, and when that is inhibited or that enzyme is absent, then we have trouble, and that results in TTP. So we've used an engineered microvessel system to study this, and this was described. Ying was the first author in a paper that we published a couple of years ago. And essentially, these vessels are made in a collagen 1 matrix from a form, and the vessels can be made in essentially almost any geometry that one wants. The endothelial cells that are seated into these channels in the collagen make junctions. They make tight junctions, overlapping junctions, and adherence junctions, and they have good barrier function. So we can take these vessels, and in this case, I have a vessel that has a geometry that looks like this, and we can perfuse blood through them. In this case, the blood has fluorescent labeled platelets. And so you see that when we put this blood through there, you get a little bit of platelet adhesion to the walls, but those platelets do not accumulate on the walls. They bind, and then they are released or embolized off. So we have a very different situation if we activate the endothelial cells in the vessels, and these are from umbilical veins, human umbilical veins. We have quite a different situation here where we've activated the endothelial cells with 4-ball myristate acetate. Now you see that there is a lot of accumulation of platelets on the vessel wall. The platelets continue to accumulate, and you get accumulation, then embolization, but eventually this can go on until some of these vessels are occluded. Now, this is whole blood, so it has the intact enzyme. If that enzyme is not there, what can happen is the vessel can occlude. And what we get when we look by electron microscopy is that we have these platelets attached to these strands, and these strands are essentially strands of self-associated von Willebrand factor. Now, I mentioned that von Willebrand factor is a multimeric protein, and in plasma we do multimer gels, and sometimes we can obtain gels that show up to 16 bands. Now, 16 bands, each band is the addition of one protomer, which is about 500,000 Daltons of molecular weight. And it's been estimated that those protomers are about 120 nanometers, so if you put 16 of them together, you'd get a pretty long molecule, 2 microns, which is about the width of a platelet. But in fact, we see really, really enormous molecules, and the reason for that is that the von Willebrand factor will self-associate. Here you see, in a straight tube, in a vessel that approximates a straight tube, flow from left to right, that strands secreted from, so the von Willebrand factor is secreted from viable Pilati bodies, and a bit remains attached, and you see that these strands will come together to form thicker strands. Here you see from adjacent streamlines strands going together. If the vessel is not a straight tube, now this is a bigger vessel of about 500 microns diameter, then you get stranding along the wall of it, and the strands follow the direction of flow, and they accumulate most where the shear stress is highest. So here you see that this is essentially following the flow, and when you get a sharp turn here, that most of the accumulation is on the inner corner of the turn. And here now we have a smaller vessel of about 300 micron diameter, where all we've done is activate the endothelial cells. No blood has been perfused, so all of the von Willebrand factor that we see is coming out of the vessel wall. And you see that this von Willebrand factor will self-associate into strands that follow the shortest distance through this vessel. It's as if you had put a line through it and stretched the line so that, in fact, you had the minimal length that would go through the vessel. And these can go on indefinitely. So here we have a section of vessel that's approximately 5 centimeters long, and there is one continuous strand. Again, there's no blood, there's no fibrin. This is just von Willebrand factor coming out of the vessel wall. And so what we've learned is that what leads these strands to accumulate, or what favors their accumulation, is shear stress and flow acceleration. So what we have here is a grid vessel where we perfuse from the bottom, and here's the outlet. And we've activated these vessels, and you see that there's accumulation of strands at the inlet. Here's where shear stresses are very high. Shear stresses are very low here because we have a very large cross-sectional area, and as one approaches the outlet, the flow has to accelerate, the shear stress goes up, and now we get strands that are oriented in the direction of flow and thickening as one approaches the outlet. And here's just a higher magnification of the outlet, and you can see that there are strands basically going in the direction of flow. There's a region right here that has quite a complex flow pattern, as the flow is coming in this direction, in this direction, and in this direction. And if you look in higher magnification of that region at what's happening to von Willebrand factor, you see that essentially you've made something that looks like a spider web there, that when blood's going through there, they can capture blood cells. So it turns out that shear stress is quite important for this phenomenon, but even more important is flow acceleration, and that's called elongational flow. And here is just demonstrated from a recent review by Tim Springer that at 2,500 inverse seconds of laminar shear stress, one gets flow acceleration, or one gets unfolding of the model polymer, and that unfolding occurs at much lower forces in the presence of elongational flow. Here's a vessel in which we've either perfused at the top normal plasma after we activated the vessel or plasma from a patient with TTP. They lack the activity of that enzyme, ADAMTS13. In the presence of normal plasma, one sees von Willebrand factor on the wall, but there's no accumulation of strands. What we really see actually on the wall are these little dots, which are essentially like stumps that are left over after the ADAMTS13 have come along and mowed away the strands. In contrast, in TTP plasma, which lacks the enzyme, you see massive accumulation of von Willebrand factor on the walls here, and in some cases even starting to occlude the vessel, and you see complex patterns of net-like formation on the vessels. So these strands of von Willebrand factor, what they do and why they're bad is because they capture platelets. And just by themselves, by capturing platelets, they can occlude blood vessels, and here the platelets are labeled pink, and you can see that in an activated vessel with ADAMTS13 inhibited, you get a lot of accumulation, particularly in the inlet to this grid vessel. But in addition to capturing platelets, we also show here that there's a lot of capture of erythrocytes. Red blood cells are captured. And of interest, that capture, if one inhibits the binding of the platelets to the strands, you still have the strands shown here in green, but you don't have platelets, and now that you don't have platelets, you don't have red cells. So we think that this has a lot to do with pathology in certain syndromes, such as TTP and possibly also malaria and sickle cell disease. In addition to those two cells, platelets and red cells, this shows that in regions where you capture a lot of platelets and you have a blocked vessel essentially that's full of von Willebrand factor, that you not only get platelets labeled here with CD41, but you get a lot of leukocytes, polymorphonuclear leukocytes and monocytes. So the von Willebrand factor can self-associate not only by the contribution of the VWF from the endothelial cells. There's also, von Willebrand factor is also a blood protein. It circulates in blood. And in this case, what we've done is we've perfused fluid phase von Willebrand factor over a transluminal strand of VWF, and you can see that the fluid phase VWF will bind on the upstream surface of this strand, further contributing to the thickening of that band. If you don't stop this, then you get trouble. So we found something that can actually prevent this by accident. And the reason that we found this by accident is because we work with purified von Willebrand factor. If you purify it too much, you lose it if you leave it in a tube. But we found that there was something in boiled plasma that could protect the von Willebrand factor. And to make a very, very long story short, that substance is ApoA1, which is the apolipoprotein of high-density lipoprotein. And that can prevent the self-association of von Willebrand factor under tubes, and then we also studied in a vessel. So in this case, we activated a vessel to secrete von Willebrand factor, and then we perfused a solution that had ApoA1. You can do that with ApoA1 or HDL. What we find that the ApoA1 will decorate the strands way better than the VWF antibody does. I guess it prevents antibody from binding. And if then you perfuse fluid phase von Willebrand factor, which is at the bottom, you get no association. And the consequences of this are that there are fewer platelets that attach to that vessel, and that's shown here. If there's no ApoA1, you get a lot of fibers of von Willebrand factor, and those fibers bind platelets. In the presence of ApoA1 in an identical grid vessel, you still get a few fibers accumulating, and that's probably because we didn't have the ApoA1 in there all the time. And pretty much the amount of platelets that adhere are proportional to the amount of fiber formation. And we've looked at in some of these disorders that have high von Willebrand factor, at the ApoA1 levels. Here's normal. In sepsis, we have a marked drop in ApoA1 and HDL, in TTP as well. And here we show that the concentration of very sticky forms of von Willebrand factor is highest when the ApoA1 antigen is lowest in these disorders. So just to say a couple of other things about other uses of these vessels, I have about 30 seconds. We can use them for angiogenesis. Here's showing angiogenic media. They make leaky vessels. Here we have angiogenesis induced by perivascular cells. Those are not leaky. We've also studied the effects of platelets on barrier function. They can restore barrier function. Here's a leaky vessel that was incubated with platelets overnight, and it became non-leaky. We've also used it to simulate some tissues, and that tells me I'm done. And here we've put CD41 cells. They attach to the wall. They actually change the character of the endothelial cells to make holes. They put through proplatelet processes or these burst-like platelet formation. So to summarize, we've developed this versatile system of ex vivo vessels where you can study all the things that I just mentioned, including thrombosis, which has not been mentioned here and I think is quite important. And this system is good because it allows modification of flow and vessel geometry, assembly of multicellular systems. You can monitor it in real time, and you can do sophisticated imaging. There are lots of challenges because this is a laborious and highly specialized technique, and it has low throughput. Nobody likes to hear this. The gaps in terms of thrombosis, I say that we still have to more precisely define the role of von Willebrand factor in platelets in a lot of these diseases of microvessel occlusion. But there are lots of opportunities in that we can use all human components. We can control the components and the timing of their use. We can be reductionist or we can not be reductionist. And we have the ability to use cells from patients or deficient mice. So I've already mentioned two of the people. I want to mention one other, and that's Dominic Chung, who did the HDL work. Thank you.
Thank you. We have time for a few questions. I have one, and the system that you showed us is sort of a purist system, isn't it? It's using purified von Willebrand factor.
The von Willebrand factor is coming out of the endothelial cells that we've seeded in it.
Yeah. So if you put blood through the system, can you make one comment about thrombosis and fibrin deposition, and also what's the viability of the endothelial cells in that setting?
The endothelial cells are more viable than they are in a planar culture. And we don't know how long you can culture them, but they seem to like to grow in circles rather than in flat planes. In terms of fibrin, that's a problem. And we intentionally anticoagulate the blood, but we are now developing a system where we can induce a fibrin clot within the vessel, but we still don't know. We're very interested in studying contact activation of blood.
Thank you. Yes, please.
There was a suggestion that actually these long strands of von Willebrand factor may be important in shearing RBCs and be part of the TTP pathology. Have you tried to replicate something like that in your system?
We have, and we see that. I just didn't have time to show you. And we actually see, in some cases, the red cells hang up and get sheared right by the strands. I just want to mention that Dr. Mayadas did some of the really great early von Willebrand factor work showing how it polymerized. I love those papers, by the way.
Thank you.
Please.
Yeah, what kind of anticoagulant you use for blood perfusion?
Generally citrate, but you can use heparin or Hirudin or whatever else. But generally,
that means when the BWF stream formation is calcium independent?
It's calcium independent, yes.
Thank you.
Okay, thank you. Please.
The self-association is calcium independent.
Nice talk. If you use ApoE1 Milano, which has been shown to be protected, you have the same effect, or is...
We haven't used it, but I think it'll work. We're trying to actually get fragments of the ApoE1 and see what's doing this, but it works if it's HDL or it works if it's ApoE1. I think Milano will probably work. I think we should try it.
Thank you. Mark?
Would you like to comment on the relationship between circulating von Willebrand factor that we can measure clinically, and on the one hand, and these strands, these von Willebrand factor strands that you've demonstrated so nicely?
Well, what we've observed, if you don't have the enzyme and you induce Weibel-Piloty secretion under flow, is that a fraction of the von Willebrand factor is released. Another fraction remains on the surface. I don't know what's the precise relationship, but what you expect would be that in those syndromes where you have a lot of vessel wall von Willebrand factor, it would be reflected in higher levels and bigger multimers of von Willebrand factor in the blood.
Thank you. Short question, please.
I just am wondering about the in vivo relevancy, because when I think of thrombosis in vivo, I think of a thrombus forming in a low shear space. You know, branch points, low shear, turbulent flow, rather than the high shear that you're talking about.
So we have in the blood two types of polymeric molecules that are involved in the adhesion of platelets. One is von Willebrand factor, which is a preformed polymer, and that one seems to function almost exclusively in high shear stress. The higher the shear stress, the more it'll self-associate and the more platelet accumulation you'll get. So that's why you get it in pre-capillary arterioles and capillaries. The other polymer is fibrin, which isn't preformed and forms and actually can't form at high shear stress. So it's kind of an interesting thing. So what you're talking about is the formation of a fibrin thrombus that's generally rich in red cells and does have platelets, but it's not dominated by platelets.
But in some acute coronary syndromes, I think the thrombus is platelet rich.
It is platelet rich, and it has fibrin, and it has von Willebrand factor.
Very, very short question.
We see a lot of things in vitro that does not happen in vivo. So I was wondering how relevant is the system for patients with thrombocytic purpura or other related pathology.
I agree with you. Well, we also see some bad things in vivo that this mimics, like TTP. I mean, actually, TTP is the reason that I went into hematology, because it was such a dramatic phenotype. So what we're trying to do here is we're trying to make this system where we can understand biophysical events that contribute to those things. I think that what we find is we've found, for example, I think already with this in vitro system, why thrombi accumulate where they do in TTP, the high shear stress regions. Two, we've figured out why in every one of those disorders where you get massive release of endothelial von Willebrand factor, there's low HDL, and it happens very fast. We think that's a sequestration event. So we're using this to help us understand the in vivo. Obviously, we have to go back and correlate all our artifacts with things that actually happen in vivo.
All right.
We're going to carry on for the panel discussion.